All right, hello everybody. This is AP Bio Unit 6. The last two sections, which I think are 7 and 8, I think. I could be wrong on that. Let's just go ahead and go with it. Yeah, topics 7 and 8. So, I'm going to talk about mutations, which is something that I'm sure you've learned before if you took biology here at our school. We'll try to do an electrophoresis gel here in class too. I have to make those gels. If you want to help me out with that, I could always use an assistant. If you have a free period, you're welcome to come up and help me get that stuff ready. If that's your thing. Can you people at home see my lecture and all that? Just want to make sure. If I could get a confirmation in the chat, that would be great. Or not. Anyone at home? Can you tell me if you can see my lecture notes, please? Yes, thank you. All right, let's go ahead and start then. So, with mutations, which is what we're going to talk about first, mutations are any change in genetic material that can alter phenotypes, okay? That phenotype alteration may not always be as obvious as yellow seeds versus green seeds. It could be a tiny change in an enzyme, okay, which may not always be a visible phenotype. Now, mutations are the primary source of genetic variation. Without mutation, you would have no evolution. So mutation rate has to be at a certain level for evolution to happen. If mutation rate would go way down, evolution would slow way down. But we don't want mutation rate so high that it produces insane levels of non-functional proteins. Now, mutation changes can be very small, so one single base could be inserted or deleted or substituted, okay, like hitting the wrong key on when you're typing, or not hitting a key, or hitting an extra key. Or they can be large-scale chromosomal changes, okay? This would be like putting a whole extra page in your document, or forgetting to put a page, or doubling the page, or something like that. So this unit sets up really nice for evolution, which is our next unit, because obviously, like I said a moment ago, no mutation, no evolution. Okay, let's move on and talk about small-scale mutations and how they can happen. So with small-scale mutations, we're going to call those point mutations because they happen at a single nucleotide, okay? And there's a couple ways those can happen. You have ATGC, and one way that can happen is to switch one of those bases with another. Okay? And there's a couple different results, okay? Now, if you remember that little chart where you take the 3 letters and you pick out the amino acid, there was some redundancy. So sometimes there are silent mutations where you get the same amino acid. Okay? Oftentimes the first 2 characters are the ones that matter, the first 2 bases. And some of the amino acid, it doesn't even really matter what the 4th base is. Okay? There might be different nucleotide sequences, those triplet codons that code for the same amino acid. So a mutation might really not do anything at all. Okay? That would be no real effect. Then you can have a missense mutation where, if you remember back at the beginning of the course, or this unit anyway, I talked about a sentence like the fat cat ate the rat. Those are all 3-letter words. This would be like changing one of those words, okay? Say, the cat ate the rat, like if you put it in again, or you could say the fat ate the rat. That doesn't even make sense, okay? Another pretty severe mutation would be changing a codon to a stop codon. So that's going to make the protein really abbreviated. So that might just be like the fat, okay? And then a stop codon comes in and we don't get the rest of the sentence. And in actuality, we don't get the rest of the protein. It's not going to fold up. It's not going to result in any, most likely, not, it's not going to result in anything useful. All right, let's take a look at a diagram of this. All right, on the left side, we have a, let me grab my iPad so I can use my pointer. Hopefully I have a little bit of charge on it, which I might not, which I don't think I do. That's what happens when I leave it in the car all weekend. It gets cold and it doesn't want to turn on. All right, I might just have to describe it instead of using my laser pointer. I'll let this charge for a minute here. Maybe we'll get some usefulness out of it. Oh, it's starting up. Okay, so look on the left side here. The original DNA level codon is TTC, okay? TTC. So when you see that TTC, if you went through the chart and decoded it, you would know that that would be the protein, the amino acid lysine, okay? So let's look at a silent mutation, and I got my iPad working. It's just a little sleepy. The silent mutation is changing the C to a T. That changes the mRNA to AAA, but that A doesn't really matter because both of those code for lysine. So this right here is a silent mutation because it's the same amino acid and the protein's going to be exactly the same. So there's more than one way to code for lysine, and this just happened to be a silent mutation because both AAG and AAA code for lysine. Okay, there are also the nonsense mutation, and that would be coding for ATC, which changes that A to a U, and you get the stop codon, which is going to cut the protein short, and it's probably going to be, like the description, nonsense. All right, missense, there are two ways that can happen. It has to do with the polarity of the amino acid, so you don't have to know that, but the important thing to know here for you is that this would change the actual amino acid, and the protein probably would be non-functional or limited in function, and that could 99% of the time, probably even more than 99% of the time, mutations are not useful, okay? Now, just current events, we have the coronavirus and its variants. The variants are useful mutations, okay? When something like a virus is copying as many times as it is in your body, okay, there's more chances for mutations. So how are we going to beat this, okay? One of the ways we're going to sort of shut this down is by getting people vaccinated, and the mutations rate should slow way down, okay? This happens to the flu virus. That's why you need a different flu virus every year. It looks like that could be something that we see with the coronavirus. You might need a new update to it every year. So some of the mutations on the coronavirus have led to these variants. I think B117 is one of them. They're calling the UK variant. The spike proteins on the outside are still the same. So the vaccine, that's what the vaccine teaches your body to recognize COVID by looking at the outside package. The inside is what has mutated a little bit. So far, we think that the vaccine is going to still protect us, okay? There are some people working on a booster for the new variants, okay? What could the mutations do? Right now, what kind of tissue does the coronavirus infect? The epithelial tissue of your respiratory lining, okay? What if it mutated to infect a new type of tissue? Nervous tissue, cardiac tissue, skin tissue, muscle tissue, okay? On that list, I think nerve tissue would be the most scary, all right? Because that's control of your body, all right? You'd start having stroke symptoms. You'd start probably having seizures. It's turning lung tissue pretty scarred up. Lung tissue is made of epithelial tissue, and it regrows rather well, okay? Nervous tissue does not regenerate very well, so that would be like a one-way ticket. The good thing about viruses that are super deadly, if they kill off the host before the host spreads it, the virus doesn't spread very well. A virus's goal is not to kill you, okay? If it kills the host, it's going to shut down its reproductive method, okay? Some of the hosts may die, but if most of them live, or they live long enough to spread it, then it will be maintained. So we really want to get ahead of these mutations, okay? Right now, in society, many parts of the country are like, we're done with this, no mask, do whatever you want, we don't care. That is a dangerous game right now. I'm vaccinated, I still have to wear my mask when I go out in public, okay? Remember, the vaccine doesn't make you bulletproof. It makes it so when you get it, it's not as severe, okay? So I still have to wear my mask so I don't become a spreader. And because I'm vaccinated, I might be a silent spreader, okay? I went out to a restaurant on Sunday and I see more people unmasked than I've ever seen before. I feel like people are kind of like, I'm just trying to tell you, be careful, because if we don't beat the variants, okay? If the variants, they're more infectious, they're more deadly. If we don't get vaccinated faster than the variants, we could see another spike. Okay, let's look at some pictures here that explain small-scale mutations, all right? Let's say we have this change right here. We see this right here, and we insert this right here. Everything after that is going to be different, okay? So that's a small-scale mutation where it's called a frameshift, okay? A frameshift mutation is usually really, really bad. Why? Because everything downstream of that mutation, where we add or take away a letter, is going to be messed up. Everything. And that can happen two ways, by insertion or deletion. So know that insertion or deletion will result in a frame shift. Now the good news is you have lots of proofreading enzymes to avoid mutations. One of the things about copying viral DNA is they usually don't have as much proofreading, so they can have a much higher rate of mutation. And they reproduce on this really large scale, so that can also allow for a higher mutation rate. All right, looks like we're ready to move on. Let's talk about large-scale mutation. They're going to happen in a totally different way, okay? These are happening on the chromosomal level, okay? We've talked about these already when we talked about cellular division. Non-disjunction is one of the, that's still considered a mutation, okay? This is when chromosomes do not separate properly in meiosis, and we get things like Down syndrome or trisomy. There's different trisomies, but this is the most well-known one. Okay, we can also get things like translocation, okay? This is when, let's make part of this blue. This is when maybe crossover is going to maybe link this up way over here instead of a section for section. We could end up with an X that's got a little bit of extra, and this one right here would be a little bit shorter, okay? So you might have something like that. These are usually fatal to the offspring. So most cases these babies will miscarry, or they will not even result in, the cells can't even undergo fertilization. Okay. Inversions would be an entire section of DNA is reversed. If you invert something, you turn it upside down or swap it. And duplication is where you can put a section in twice when it shouldn't be there. And remember, this is a segment. This is a big, long set of bases. These are large. All right, everybody comfortable with me moving on? Getting some yeses over there? All right. Deletion, that's where a big section is obviously lost or deleted. All right. So let's just touch on evolution, which is our next unit. So we're going to kind of like lay a little groundwork for it right here. Anytime you get a mutation, they're subject to natural selection. So what does that mean? All these genetic changes can sometimes, usually doesn't happen, but sometimes they might enhance the survival and reproduction of an organism. So maybe the mutation leads to an enzyme that's more efficient. Maybe the mutation leads to a trait that allows for better camouflage or better whatever, okay? Anything that allows them to survive and reproduce, okay? That could be, something way down on the cellular level that's hard to see, or it might be a phenotype that's easy to see, like fur color, or maybe a reproductive strategy, or something along that line. So if whatever it is good, okay, then there's a really good chance that trait will be carried on. Okay, I've told you several times, my wife and daughter both have this gene for a blood clotting disorder where their blood clots too easily. And we went and saw a specialist in that area, and he said what likely happened was someone in my ancestry and my wife's, Northern Europe, had a lot of kids with this. He had the mutation and he had a lot of kids. It made blood clot easier. Well, when we were out there stabbing each other and fighting to survive a lot more, in warfare and in just day-to-day hunting and gathering, having blood that clotted a little bit faster than everybody else was a good thing. Now that we just lay around and sit on, computers all day, it's not such a good thing. Okay, it can lead to clots that travel to your brain and kill you. All right. And that's what actually happened to my wife's father. He died of a blood clot in his mid-50s. It went to his lung. All right, so let's look at another way that we can increase genetic variation. Prokaryotes, they can mutate as well, but they can also do this thing called horizontal gene transfer, okay? This is the closest you can get to bacteria sex, okay? If there is a mutation that's beneficial to survival, okay, in prokaryotes, they can also transfer genes back and forth, okay? And we talked a little bit about this in one of the assignments I gave you, where the Griffith would boil and kill the agent that was causing disease, but the bacteria would pick up that agent and still kill the mice. So we've done a little assignment on this already. So there's a couple ways this can happen, okay? We can do transformation. That's where you take up the DNA from a nearby cell, okay? We can do transduction. Viruses will actually change your DNA sometimes. They will inject their own little statement into your genes. We can do conjugation. That's cell-to-cell transfer. And then we can do transposition. That's DNA segments moving between DNA molecules, okay? So that's sort of like an internal thing. Now, we are going to try to do a lab, and most of the time we pull it off, but sometimes we do it and it's not successful, where we do bacterial transformation. Now, instead of from a nearby cell, we're just going to cheat and just surround the bacteria by little plasmids of DNA that we introduce to the environment, and then we do things to the cell. We get it cold, we get it warm, and that kind of opens up its cell wall, and hopefully we get that little ring of plasmid inside of that bacteria. And when we do, we transform that bacteria to have a new genome, okay? It's going to now have a genome that it's going to do what we want it to do, and that's make this jellyfish protein that glows in the dark. Also on that plasmid is antibiotic resistance, which will do a little experiment where we put a little antibiotic on our plates to see if the cells can grow around it or not. See if the bacteria can grow around it. So very, very cool lab, okay? Stuff that doing this even when I was in college was like, whoa, nobody in college, unless you're a grad student or PhD, you're not really going to be doing that. But now we can do it in a high school classroom. Okay, so that was section 7. I'm just going to quickly run you through section 8 right in the same lecture. That'll be the end of unit 6. So there's lots of biotechnologies out there, okay? We're living in an age where biotechnology is super important to developing this vaccine. We've never had a vaccine that quite works like this one does. So, I like to think I grew up in the electronic age. I feel like you guys are growing up in the biotechnology age. The thing is, people are starting to be able to do this stuff in their garage. There's a really cool Netflix series on this. I don't remember what it is, but it's all these people trying to hack their own DNA. They're injecting themselves with these things that modify their genome to make them grow more muscles. It's really controversial about like who should have access to this, okay? There's a lot of bioethics and a lot of sort of questions about, is it okay to just let anybody go out there and use these tools to do whatever they want to do? There could be a lot of implications. So we live in a world where DNA and RNA can be manipulated through genetic engineering. Okay, to make these vaccines we're using, we had to make a little piece of mRNA that coded for exactly what we needed to code. And so we know how to do this now. We can edit DNA and RNA just like you edit a document on your word processor. I mean, it's a little different, but so that right there is a huge one, okay? Now this one's been around for a long time, and we're going to try to do this one in class as well. Gel electrophoresis. You probably heard of this one, okay? In this one, you make a block of gel, okay, and you basically have a mold. You pour this gel in there, and it solidifies just like Jell-O, but you make little wells or holes on the end. You put this thing called a comb in the gel, and when it gets solidified, there'll be little pits, okay, called wells. You take a pipette and you inject a small amount into each well, and then you hook it up to electricity, okay, and you run electricity through the gel. And you can load DNA into it, and DNA is negatively charged, so things in the wells, you put the wells on the negative end, and the DNA will move toward the positive end. Well, there's little tiny holes and pores in the gel, okay? And the smaller fragments move farther through the gel. Now, you can do this with other stuff too. You can take the candy dye on Skittles, dissolve it, and put like all the purple in one and the yellow and the orange, and different parts of the dye will separate out. I like doing it with the candy dye better than the DNA fragments because you can see it so clearly. Okay, you'll see like a purple divide out into its base, the colors it was mixed from, and you can tell which dye molecules are bigger or smaller. So gel electrophoresis allows us to do a couple different things. We can see if two DNA samples match. Okay, so you can kind of do some forensics with this. Like, hey, we got to get a blood sample from the crime scene, and we're going to see if it matches the suspect. Now, the tests are actually more sophisticated than that now, but this is something you could do. Okay. PCR is basically a DNA copying machine. Okay. That stands for polymerase chain reaction. Okay. You've probably heard of polymerase before. And so in this, we take a segment of DNA and we make millions and millions of copies of it. Well, why would we do that? Sometimes it's very difficult to, hey, we're going to study this gene. We need lots and lots of copies of it. Hey, we're going to run this gene through electrophoresis gel. Well, you can't just put one piece of DNA in there. You got to put thousands. And so there's a lot of times in biotechnology where we need lots and lots of copies of a gene to study it. One of the early COVID tests was a PCR test where they would take a sample out of your nose, PCR it, and get the amount of viral DNA up to a level that was high enough to detect. Okay? So that's... Huh? They're requiring a PCR test for airlines? Okay. So if you hear someone say, I had to get a PCR test, because I believe it's more, the results are better. It's more accurate than some of the other tests, okay? Which is why it's sort of a higher standard. In Ohio, the governor DeWine was supposed to meet with Trump, and they announced that, he can't do it because he has coronavirus. He had a false positive with the rapid test. This test takes a little longer because we have to go through this process. And everyone thought he had it. He took another more accurate test. It was a PCR test. And we got rid of his false negative, okay? All tests have false negatives and false positives, guys. None of these biological lab tests that you get are usually 100% right. You go into surgery, they're usually going to give you 2 or 3 blood tests just to make sure if they have to give you blood during surgery, they're not going to give you the wrong blood type. So they like to have more than one test before they start doing life-risky, risky procedures. So basically this amplifies segments of DNA and makes more and more and more copies of them, and it can do this very, very quickly, okay? You start with 1, you go 2, 4, 6, you get this geometric increase or exponential increase. And in a couple, in a half hour, you can have millions of copies of a gene. And this is what you put in those electrophoresis gels when you pipette it into those little holes. Okay. Now, DNA sequencing, which I'll show you something about my DNA sequence after we're done with this lecture here. DNA sequencing can happen in a lot of different ways, okay? Don't really care that you know all the different ways it happens. There's little transistors that can detect bases. It's really a cutting edge kind of thing. We did that BLAST lab where you took a DNA sequence, typed it into a database, and identified a disease, okay? All of that has happened in various ways. There are these chips, and when I say chips, I mean computer chips that are getting faster and faster and faster and faster at analyzing DNA, okay? Back in the 90s, if I said I wanted to analyze your complete genome, you would have been paying, thousands and thousands of dollars for that to be done. Like all things, the price of technology comes down. You can now order a kit from 23andMe, spit into a little test tube, close the lid, send it away to them, and they'll analyze your DNA for sometimes they have sales where it's $100. okay? So these chips have gotten cheaper and cheaper and cheaper and cheaper. And what does this allow us to do? There's lots of applications for this. One is medicine, just figuring out what causes diseases, okay? Figuring out what causes different phenotypes that might cause you to be the carrier of some horrible genetic disease, okay? Before we were able to do this and test for these, there were genetic counselors that would say, okay, this disease runs in your family. If you have kids, there's nothing we can do to change the odds. Are you prepared to bring a kid into the world with a 25% chance that it's going to have cystic fibrosis? Okay? So now we can actually sequence the gene. We can do a genetic analysis on the embryos and figure out, is this going to be a gene that is this embryo going to have the gene for this terrible disease? We can screen the embryos, so you can do in vitro, check all the embryos, discard all the ones that are carrying the disease, and just implant the one that's healthy, okay? There's ethical questions involved in this as well, okay? So, do we do it for disease, or do we start doing it for I want my kid to ace the SAT and to be an Olympic athlete, so pick out all the good ones. Is that okay? Some people would say yes, some people would say no, some people would say it depends, okay? So we talk about health, testing, identifying, and even treatment, okay? We can use this to preserve endangered species, okay? We get the gene sequence of something that's endangered, and maybe now we can edit that gene, reassemble that gene if they go extinct, okay? You've all seen Jurassic Park. We're not quite there yet because we really can't just put frog DNA in the stuff that's missing, okay? That's how they said they got away with it in the movie. Some other areas are forensics, okay? We can take blood samples or tissue samples or whatever under fingernails at crime scenes, and we can show that that's your DNA at the crime scene, okay? We can study evolution this way, okay? We can say, hey, the DNA in a chimpanzee is 97% the same as in a human, so they must be very closely related compared to the DNA in a human and a orangutan. So which one's closer related? So we can do things like that. In the coronavirus, we were very quickly able to figure out if this was a transzoic virus, okay? We take and analyze the DNA in the virus and we say, yep, it came from bats or it came from this or that. That can be very important. So when you hear things like bird flu, we're looking at viruses transferring from species to species. So there's lots and lots of applications for this. Okay, genetic engineering. There's this stuff called golden rice. A lot of people that have a largely rice diet don't get a lot of vitamin A, and there's a lot of blindness in some of these places that don't have as developed as food systems as other parts of the world. We can genetically put in a sequence of genes that makes rice make vitamin A, and it pretty much eliminates vitamin A related blindness due to vitamin A deficiency, and it's genetically modified, which some people don't like. You'll notice as we get into biotechnology, the ethical questions really start coming up more and more often. There's a really good science fiction movie out there called Gattaca, which is, by the way, spelled with bases of DNA. You've seen that? Yeah, it's shown a lot in school, okay? Most people have seen it already, so I don't really show it. It has Ethan Hawke in it, and in this science fiction future, they kind of segregate people based on their genetics. And he was born by natural conception, and he's considered like an outcast. He has to wear contact lenses, which was, because they eliminated bad eyesight, okay? And he was trying to be an astronaut and go on this space mission by not being genetically modified. So it made two classes of people. People who were genetically screened and modified versus just natural people. And natural people were considered like second-class citizens. All right. Lots and lots of cool stuff becoming more and more accessible to society. So this is probably going to be an area of technology that really explodes, okay? In my day, like I said, it was electronics. I think for you guys, biotech is going to be huge. All right, let me go ahead and stop this recording. We'll talk.